So it's my pleasure to introduce our next speaker, Martina, who's professor of neuroscience at Northwestern's own Feinberg School of Medicine. Dr. Martina has investigated the functional and pharmacological properties of ion channels for nearly 25 years. And today he'll be speaking on cell type specific alterations of cortical excitability in a model of familial ALS.
Okay, so thank you for the introduction. Thank you for the organizers for giving me the opportunity to present in this venue. I'm a relatively newcomer to the field, so it's a great learning experience. As said, I will talk about cell-specific alterations, and we'll look in excitability and inhibition. And we move now from the spinal cord to the brain, to the motor cortex in particular. And cortical hyperexcitability is actually a major feature of ALS. And you can see here, if you do a PubMed search of ALS and motor cortex excitability, there has been a dramatic increase over the past years in the number of papers you can find about this theme. And it's very interesting because numerous studies show that the cortical hyperexcitability is actually something that is found in ALS patients of any known etiology. All the different types, they all seem to share this property. So it might have a really important role in the pathogenic mechanisms. And why the model we use? The model we use is this Alsin knockout model. And this is a protein that was found to be associated with ALS in two studies in 2001. They were back-to-back in Nature Genetics. One from Tipu Siddiq, that was a member here of the ALS Center here. And this study examined families, one in the Middle East, one in Africa, that exhibited a early onset ALS, and they did a genetic study. And they found that there was this gene, ALS2, that was mutated in both cases. So then people found what is the protein that is the product of this gene. The protein was named alsin, and several studies started to characterize the effect in cell cultures, in vitro systems of this protein and how it might induce pathogenicity. And one of the things that they found, which I think is quite interesting, is that there is a correlation between this protein and other ALS models. For instance, these two studies that I'm showing here, they identified mechanisms by which actually alsin decreases the normal alsin, not the mutated, right? Decreases the negative effect of SOD1, which was the very first genetic model of ALS. So we can think that this mechanism, on the one hand, is specific because it's this one gene that is mediated, but on the other hand, may have a more general importance for ALS in general, because it interferes with other pathogenic processes as well. And one of the persons that has been pushing ahead the research on this alsin mutation is actually Hande Stindler, that made a mouse to study this mutation specifically in upper motor neurons. And here we come to what we do in my laboratory. So our experimental approach was, and all experiments I'm going to present today were performed by Xiaomil Day, a postdoc, in talented postdoc in my laboratory. And what Shamil did, he took advantage of the mouse that Mukesh Gautam and Anders Dingler developed. And this mouse is a mouse in which EGFP, so a fluorescent reporter, is expressed in the UCHL1 expressing neurons, which in the motor cortex characterizes the motor neurons that project down to the spinal cord. So you have these slices, as the image you see there, this image of a motor cortex slide, and slice. And there are cells side by side, and the GFP positive cells are actually the cells that project down to the spinal cord. And the cells that are just side by side, but they are negative, they are the commissural cells that project to the contralateral side of the cortex. And they also did cross these animals with Alsin-KO animals. So now we have wild type animals or Alsin-KO animals, and in both cases, they express the GFP selectively in these neurons. And therefore, we can then either use an electrophysiological approach, and this was all done in 1-month-old animal, pre-symptomatic, right? These animals at this point have no symptoms. And we can do recordings and look at excitability of the cells. But what Shamil also did, he put some tracers inside the pipette, so it could then really have a post hoc anatomical reconstruction of the cells, and so that we can do an anatomical analysis of the patched neurons. So the first thing that he did was a very simple approach. He wanted to make sure that these two cell types that are characterized by either the expression or the non-expression of GFP indeed are functionally different populations of cells. So he did a very simple test. He patches the neuron, he injected negative current injection, and this can give you already many really important informations about the properties of the cell. So the first thing, obviously, you can measure is the difference between the baseline and the total in the voltage response elicited by the current injection. By simply applying Ohm's law, which states that the resistance is equal to the current divided by the voltage, we know the current is what we inject. We know the voltage is what we measure, the voltage response, so we obtain the resistance of the cell. And the input resistance of the GFP positive and GFP negative neurons in wild-type animals, that is really different. You see a very clear difference there. But this simple experimental approach gives another type of information here. You can fit the exponential time course of the voltage response, and from basic circuit laws, we know that the capacitance is proportional to the time constant divided by the resistance. So now we can also obtain measure of the capacitance of the cells. The capacitance is a measure of the size of the neurons. And so again, you see a clear difference between the two groups, with the neurons projecting farther away being longer. As you imagine, they need bigger cell bodies, they have to produce more protein, they have to be able to sustain the nerves that go all the way down to the spinal cord. And there is also a third measure we can get here. Here is the difference between the peak response and the steady state response. This gives us a readout for a type of current that's called the hyperpolarization activated current. And also this, you see, in this case, the two cell types are really, really different. So we then looked at the active properties, the excitability really of the cells. In this case, you use a positive current injection, and this elicits action potentials. And you use synaptic blockers, so you only focus on the intrinsic properties of the cells you're studying. And you see right away from these traces that the traces in green here that represent the cells with GFP are more excitable than the other cells, than the commissural cells. And indeed, the input-output curve is different, the maximum firing frequency is different. So we have established very clearly that the two cell types are functionally very different, and they cluster actually in two quite distinct problems. So we have these two cell types. Okay, now we have these two cell types. Now we can go, and each of these cell types, we know we can compare their properties in ALS, in Alsin-KO mice versus controls. And so the first thing, Xiaomil looked at the commissural cells. So the GFP negative cells, these are the cells that are in one side of the motor cortex that just they project to the contralateral side. And there is absolutely nothing to be said about that. There is no difference at all. Nothing you can see here, just an example of both the excitability, so the firing pattern of the cells. You see the input-output curve. There is absolutely overlap, complete overlap. And even for the negative responses, the passive, let's call cable responses, you see absolutely no difference, right? So a little bit underwhelming. But then we looked at the corticospinal neurons. So now we patch the GFP positive cells. And you see right away from this, just looking at the firing pattern, that the wild type and the KO are very, very different. What we didn't expect is that the KO are actually less excitable. We expected the opposite, but that's why you do the experiments, I assume, right? And so they are less excitable. The input-output curve is dramatically different. And the maximum firing frequency is also different. So there is two surprises here, right? The first is that in the commissural cells, there is no difference, but here there is a really big difference. And the other is that there is a hyperexcitability. The third thing is also, these animals, as I mentioned, are completely asymptomatic, and yet their motor neurons seem to be very different in their excitability. The two things that don't match, they're hard to match, right? So I realized, okay, all the experiments we've done were in the presence, as I mentioned, of synaptic blockers to look really at the intrinsic excitability of these cells. But what happens if we leave all the synaptic inputs still active as much as they can be active in a slice, right? So we did this experiment without synaptic blockers, where inhibition is intact in the slice, and suddenly all the differences between the wild type and the Alsin-KO animals disappear. There is no difference in excitation at all. Remember, here just to remind you, with synaptic blockers, there was this huge difference. So this then suggests that probably inhibition has a major role in controlling the activity of these cells. This makes sense. Okay, so we have some potential differences. The inhibition may be in homeostatic role, and the excitability, intrinsic excitability and the inhibition, they have this reciprocal homeostatic control. But at the end of the day, you have no difference in excitability, which explains the no symptoms. So if that is the case, that indeed the inhibition is so important, then we can study that, right? Because when we patch the neurons that we show here, the neurons, even in the slice, will have multiple interneurons, here represented by this orange small cell, that project to these neurons and release inhibitory neurotransmitters, GABA in this case, and inhibit the function of the cells. So by recording the cell, the motor neuron, in this case, the upper motor neuron, we can see these spontaneous inhibitory events that reflect the activity of the interneurons, the inhibitory inputs. And you can see right away that the traces look quite different in the Alsin-KO and in the control animals. And if you do a quantification of this data, you see there is no difference in the mean amplitude of the currents that are elicited in the motor neurons. But if you look at the frequency, there is a very large difference in the frequency, which is reduced in the Alsin-KO animals, which explains because you have less inhibition, so therefore you have an overall boost to the excitability of the cells that compensate the reduction in intrinsic excitability. At this point, I'd like also to make a point. The fact that we have this very specific difference in the excitability between the two cell types, and again, these are two cell types. They are, in the slides, they are side by side. They are really very close. There is no difference in distribution, right? So this, to me, suggests that probably what we are looking at it as a mechanism is something that is really cell type specific, and which means I don't think in this case, at least in the case of the upper motor neurons, that something like general inflammation is really important at this stage, because it's difficult for me to figure out a difference in input from cytokines or things like that could really affect specifically one cell and not the neighboring one. Although there could be specific receptors that are expressed, but then if there are specific receptors, we go back to the intrinsic difference. And so and then in the Osdinler lab was done with the cells that showed Mill field, they did a staining for VGAT, which is the glutamate, vesicular glutamate and GABA transporter, which is a marker of presynaptic terminals, and with Gefferin. Gefferin, as shown here in the schematic, is actually a scaffolding protein that holds together the postsynaptic GABAergic receptors. So when you have an active inhibitory synapse, you must have the postsynaptic receptors for GABA and the presynaptic terminal that releases GABA. So then, by doing the staining for VGAT, the presynaptic terminal, Gefferin, the postsynaptic inhibitory, and we have the dendrites stained with the tracer that was injected, we can look on each cell and count how many inhibitory synapses are there. And indeed, there is a dramatic decrease in the number of the inhibitory synapses on the Alsin-KO cells. So in conclusion, we show that in pre-symptomatic Alsin-KO mice, intrinsic excitability is selectively reduced in upper motor neurons. In this pre-symptomatic Alsin-KO mice, synaptic inhibitory input is also selectively reduced in upper motor neurons. And again, this selectivity suggests probably a genome-specific effect. And that native excitability of upper motor neurons is unaffected in these animals, which is in line with the fact that they are pre-symptomatic. And then the question that was already asked in the previous talk, which comes first? And, histological analysis suggests that decreased synaptic input and decreased excitability are linked in a homeostatic loop. But is the intrinsic excitability response to the decreased inhibition, or vice versa? That is what we are going to study, I hope, in the future. And all this that I presented as obviously would not have been possible without our funding support, and the Les Turner ALS Foundation has been critical. Nothing of this would have been done if we hadn't that fund. And then obviously NIH, the National Institute of Drug Abuse, and National Institute of Neurological Disorder that support our lab. And many thanks obviously to Xiaomil Day, who did all the work, and Mukesh and Hande provided the animal, and Christopher Quintanilla and Dylan Beatlis in the lab that did the quantification of the histological data. Thank you.
So it's now open for questions. Yes, please.
So one of the hallmarks of garden variety ALS is fasciculations, which would represent a hyperexcitability. And I'm wondering if you've been able to, even in a pre-symptomatic situation, look at motor unit hyperexcitability in the muscle itself. The second thing is, gosh, there are other genetic models of ALS, and do you find similar changes in the inhibitory pathways in them as well?
Yeah, thank you. So about the fasciculation, we did not characterize that carefully this animal. It's a very interesting point. I think it's a great point. It's something we definitely should do. About the other, so we had a analysis a few years back. We looked at SOD1 mice, and there as well there is some interesting. We found the differences are less pronounced, but there is also in pre-symptomatic stage a decrease in intrinsic excitability, which matches what we find here, and a decreased inhibitory input that was selective for some layers of the cortex. So yeah, it seems to hold true in, okay. And I think